Welcome to the Deep Dive, your shortcut to being well-informed. We sift through the sources, pull out the key insights, maybe even some surprises. Today we're digging into what you might call a hidden enemy, something that has a huge impact on a major global industry, but often flies under the radar. We're talking about Porcine Proliferative Enteropathy, or PPE. You probably know it better as ileitis. It's this really widespread bacterial disease in pigs. So our mission today is to really unpack this. We'll look at the scientific and detective work it took to figure out the cause. It's pretty unique biology, and the cutting-edge ways producers are trying to control it now. We've got some great sources for this: detailed scientific reviews, research papers, the whole nine yards.
Yeah, and what's really striking is just how widespread ileitis actually is. I mean, globally, you find it pretty much everywhere pigs are raised, on all kinds of farms, and it's often this sort of silent thief. It creates this massive economic drag, especially the subclinical forms that don't show obvious signs, which is why getting a handle on ileitis is just so critical for the swine industry.
That really paints a picture. Okay, so for anyone maybe new to this, what exactly is porcine proliferative enteropathy, or ileitis?
Well, at its heart, it's a bacterial disease, very common in pigs. It mainly goes after the intestines, specifically a part called the ileum, the lower part of the small intestine.
And you said common, but it sounds like the impact is anything but small.
Oh, absolutely. The economic hit is huge. We're talking increased deaths, more sick pigs, slower growth. They don't convert feed efficiently.
Right.
And you end up with groups of pigs that are all different sizes when they go to market, which, really impacts the farmer's paycheck.
And you weren't kidding about widespread. I saw figures suggesting over 90% of herds in many countries show exposure.
That's right. And maybe 40% or more of individual animals. It really is almost everywhere. Ubiquitous is the word.
So it's not just one simple thing then.
No, not at all. It's really more of a spectrum. How ileitis looks depends a lot on the pig's age, its immune status, things like that. So you can get these subtle subclinical infections where pigs just aren't doing well, right up to really severe, even deadly forms like necrotic or hemorrhagic enteritis.
Which must make it tough to manage.
Definitely. That variation makes diagnosis and control a real challenge on the farm. And the bug behind all this is called Lawsonia intracellularis. It's an obligate intracellular bacterium.
Obligate intracellular, meaning it has to live inside a cell.
Exactly. It can't survive or multiply outside of a host cell, which, as you can imagine, makes it pretty tricky. It infects the cells lining the intestine and triggers this abnormal growth, this proliferation. That's where the name comes from.
Okay, and finding this tricky bug sounds like a story in itself. You mentioned a scientific detective story.
Oh, yeah, it took decades. The typical damage in the gut was actually first described way back in 1931. They called it porcine intestinal adenoma then. But for like 60 years, it went by all these different names: regional ileitis, proliferative, hemorrhagic enteropathy, because everyone suspected it was infectious, but nobody could pin down the actual cause.
60 years is a long time in science.
It is. Then in 1973, there was a bit of a breakthrough. Researchers using electron microscopes finally saw these small curved bacteria inside the affected gut cells. Based on the shape, they initially thought, okay, maybe it's some kind of Campylobacter.
But it wasn't Campylobacter. Why was it so hard to nail down?
Well, that goes back to that key point. It's obligate intracellular nature. Think about it. Standard procedure is you take a sample, put it on a petri dish, a cell-free culture medium, and see what grows. But they tried that over and over with this suspected bug, and nothing. It just wouldn't grow without living cells, and that failure was a huge roadblock. It meant they couldn't fulfill cost postulates, the standard rules for proving a specific microbe causes a specific disease.
So how did they finally crack it?
The big moment came in 1993. Scientists finally managed to cultivate it not on a plate, but inside a specific type of living cell line, actually. Rat enterocyte cells. That was the game changer. Once they could grow it, they could study it properly, characterize it, and they realized, okay, this isn't Campylobacter, this is something totally new. And that's when it got its official name, Lawsonia intracellularis. It really unified the understanding of ileitis as one disease caused by this one specific, very sneaky bacterium.
Sneaky is right. So how does this master manipulator actually work once it's inside the pig? It's not just a typical bacterial invasion, you said.
No, it's much more subtle in a way. So Lawsonia is gram-negative, kind of curved or S-shaped. And yeah, it has to live inside cells. But here's the clever part. Most bacteria might invade a cell, replicate, and then destroy the cell to spread. Lawsonia does the opposite. Its survival depends on keeping the host cell alive and, crucially, making it proliferate, making it divide over and over again.
That would create its own little factory.
Exactly. It expands its own niche by making the host cells multiply. And that abnormal multiplication is the lesion, the thickened gut lining we see. Infection starts typically when a pig eats contaminated feces. Fecal-oral route is key. The bacteria then actively invade the cells down in the intestinal crypts, the little valleys in the gut lining, especially in the terminal ileum.
And once it's inside,
It's pretty sophisticated. It quickly breaks out of the little bubble it entered in, the vacuole, and starts replicating freely inside the cell's main compartment, the cytoplasm. But the real hallmark, the signature move of Lawsonia, is how it messes with the cell's life cycle. Normally, those young crypt cells mature into specialized cells that absorb nutrients, then they stop dividing.
okay.
But Lawsonia stops that maturation process. The infected cells stay immature, they stay proliferative, they just keep dividing and dividing. It's like they're stuck in adolescence, cell-wise.
And the bacteria just go along for the ride into the new cells.
Precisely. As the host cell divides, the bacteria get passed down to the daughter cells, so the infection spreads vertically within the crypt structure. All this uncontrolled growth leads to that dramatic thickening of the gut wall, which means less surface area for absorption, leading to poor nutrient uptake, diarrhea, and poor growth.
Wow. How does it actually control the cell like that?
It's believed to use something called a type 3 secretion system. Think of it like a tiny molecular needle. It probably injects specific effector proteins directly into the host cell, and these proteins interfere with the cell's own signaling pathways, essentially hijacking its controls to keep it dividing.
Incredible. So we know it's complex, and you mentioned a spectrum of disease. Let's unpack the different ways ileitis can actually look in a pig.
Right. There are sort of four main forms, though they can overlap. Which one you see depends on things like the pig's age, immunity, maybe the dose of bacteria they got, other infections.
Okay, what's the most common?
That would be porcine intestinal adenomatosis, or PIA. This is the chronic, sort of uncomplicated form. You typically see it in younger pigs, maybe 6 to 20 weeks old after weaning in the growing phase. The signs can be subtle, maybe some intermittent loose grayish diarrhea, but not bloody. The big thing here is the performance hit.
So they just don't grow well.
Exactly. Reduced gain, poor feed conversion, and you get this annoying variation in size within the group. Mortality is pretty rare with just PIA. If you were to look inside, post-mortem, you'd see that characteristic thickening of the last part of the ileum, often looking kind of folded or corrugated.
Okay, what's next on the spectrum?
Well, sometimes PIA can progress to necrotic enteritis, or NE. This is also chronic, but it's more complicated. Basically, that overgrown tissue starts to die off, it becomes necrotic. Clinically, these pigs often have persistent diarrhea and really waste away. At necropsy, the thickened gut lining is covered with this thick yellowish-gray, almost cheesy layer of dead cells and inflammatory gunk.
Sounds unpleasant. Are there other chronic forms?
Yes, there's one called regional ileitis, or RI. Sometimes people call it hosepipe gut. This often seems to be a later stage, maybe a healing or scarring stage after NE. The result is a section of the ileum becomes really thick, rigid, like a stiff hose.
Hosepipe gut. Okay.
Yeah, and that stiffness can cause blockages or slow down gut movement.
And then there are acute forms, too.
Right. The most severe acute form is proliferative hemorrhagic enteropathy, or PHE, often called bloody gut. This usually hits older pigs that haven't been exposed before, think late finishers, new gilts entering the breeding herd, young sows, maybe 4 to 12 months old. The onset is sudden and dramatic. You see profuse, dark, tarry, or even bright red bloody feces. That's called melena.
Yeah, it sounds serious.
Extremely. The pigs get pale very quickly, weak, and often just die suddenly. Mortality in pigs showing PHE signs can be really high, up to 50%. Inside, you find the gut filled with this massive clot of blood, often extending through the ileum and into the large intestine.
Wow. So PIA, NE, RI, PHE.
Yeah.
Quite a range.
And there's one more crucial one, subclinical infection. This is maybe the most common overall. These pigs show no obvious signs of diarrhea, nothing you'd easily spot, but they're still infected, and they suffer from reduced growth and poor feed efficiency.
The hidden losses you mentioned earlier.
Exactly. They take longer to get to market, make the group less uniform. It's a constant, quiet drain on profitability. So yeah, understanding which form or forms are present on a farm is really key to tackling it effectively.
Absolutely. So given this tricky range, from invisible losses to sudden death, how do vets and producers actually nail down a diagnosis.
It's usually a combination approach. You need good diagnosis to make good control decisions. Often, you can get a pretty good idea, a presumptive diagnosis, just from the farm history, the age of the pigs affected, and the clinical signs you're seeing.
Like seeing bloody gut in finishers points towards PHE.
Right. But for a definitive diagnosis, especially for the less dramatic forms, you usually need a postmortem exam, a necropsy on affected pigs. The changes in the gut itself are often quite characteristic.
And then lab tests come in.
Yes, lab tests are crucial for confirmation. Histopathology is sort of the classic gold standard that involves looking at thin slices of the affected gut tissue under a microscope. You see that telltale sign, the massive proliferation of those immature crypt cells. And you can use special stains, like silver stains, to actually visualize the little curved Lawsonia bacteria inside those cells.
Seeing is believing.
Pretty much. Another technique, also considered a gold standard, is immunohistochemistry, or IHC. This uses specific antibodies that stick only to Lawsonia antigens in the tissue, so you get this very clear visual confirmation of the bacteria right there in the damaged areas.
Okay, what about faster methods? I hear a lot about PCR.
Yes, PCR, polymerase chain reaction, and its quantitative version, qPCR, are very important tools. They're incredibly sensitive at detecting the bacterium's DNA. You can run PCR on feces, gut tissue, even oral fluids nowadays. And qPCR is great because it tells you how much bacterial DNA is there, the bacterial load.
Like a high load means active disease.
Generally, yes. Low CEAT values, which means high bacterial load, correlate well with active disease. High CEAT values might mean low-level shedding, or maybe the pig is recovering. There is a key limitation, though. PCR cannot tell the difference between DNA from a live vaccine strain and a wild field strain. That's important to remember.
Ah, okay. Good point. What about blood tests? Serology.
Serology, usually using an ELISA test, is really valuable for herd-level monitoring. It doesn't detect the bacteria itself, but the pig's antibody response to it, specifically IgG antibodies in the blood. This tells you if a pig or the herd has been exposed in the past. Pigs usually develop these antibodies about 2 to 4 weeks after infection.
So it's good for tracking exposure patterns.
Exactly. It's excellent for understanding the prevalence in a herd and figuring out when pigs typically get infected. That timing is crucial for planning control, especially vaccination. But, and this is important, serology is not useful for diagnosing disease in a sick pig right now. It just shows past exposure, not necessarily current illness caused by Lawsonia.
All right, different tools for different questions. Okay, so we understand the bug, the damage, the diagnosis. How do we actually fight back effectively, especially now? You mentioned strategies are evolving.
They really are. For a long time, the main approach was often putting antimicrobials in the feed continuously, prophylactically.
Just prevent it.
Yeah, but there's huge global pressure now to reduce antibiotic use in livestock, mainly because of concerns about antimicrobial resistance, or AMR. So that old strategy is really on the way out. Effective control today demands a much more integrated approach.
Meaning you combine different tactics.
Precisely. It's about combining good management and biosecurity with strategic use of antimicrobials when needed and critically effective vaccination programs.
Let's break that down. Management and biosecurity, what does that involve?
Basic but vital stuff. Strict hygiene, really thorough cleaning and disinfection of barns between groups of pigs. That reduces the environmental load of the bacteria. All-in, all-out production systems are very effective. Moving all pigs out before a new group comes in breaks that cycle of transmission from older pigs to younger ones. And minimizing stress is huge. We know stress periods, weaning, transport, mixing pigs, feed changes often trigger ileitis outbreaks. So anything you can do to smooth those transitions helps.
Makes sense. What about antibiotics now? Are they still used?
Oh yes, definitely for treatment. If pigs are clinically sick with ileitis, therapeutic use of antibiotics is still necessary. Things like macrolides, Tylosin is common, or plurimutilins like tiamulin, lincomycin, tetracyclines. They can be given by injection, in water, or sometimes in feed for treatment. But the key shift is away from just routinely feeding antibiotics for prevention. Besides the AMR issue, constant low-level antibiotics can also prevent pigs from developing their own natural immunity.
Right, which brings us to vaccination. You said that's a cornerstone now.
Absolutely. Vaccination is central to modern ileitis control. There are basically two main types of vaccines available. First, there's an oral live attenuated vaccine. Enterocellularis is a common example. You give this to pigs through their drinking water.
Live attenuated means a weakened form of the bacteria.
Exactly. It's alive, but can't cause disease. It stimulates a really good immune response right there in the gut mucosal immunity as well as systemically. Provides solid protection. But There's a big operational constraint. Because it's live bacteria, you have to stop all antimicrobial use for a period around vaccination, typically at least 3 days before, the day of, and 3 days after.
The antibiotic-free window.
Right. And that can be a real challenge in some herds if they're also dealing with other bacterial diseases that need antibiotic control during that nursery period.
Okay, so what's the alternative?
The alternative is an injectable inactivated vaccine, sometimes called a killed vaccine. Porcine is an example. This one is given by intramuscular injection. Being inactivated, it primarily stimulates a strong systemic antibody response, its main advantage. Its efficacy is not affected by antibiotics being used at the same time, so it offers a lot more flexibility for herds that need medication.
But it's injected, so more labor-intensive.
Yes, you have to handle and inject each pig individually, unlike water administration. So, it's a trade-off. Choosing the right vaccine, live, oral, or killed injectable, really depends on the farm's specific situation, their health challenges and medication protocols. There are clear pros and cons to each.
And it sounds like this move away from routine feed antibiotics has really put the spotlight back on ileitis.
It absolutely has. In many ways, the widespread prophylactic use of antibiotics was masking the true extent and impact of ileitis, especially the subclinical form. As producers reduce that usage, they're often seeing ileitis emerge or reemerge as a major issue, which just underscores the need for these more sophisticated, proactive control strategies like vaccination and top-notch biosecurity.
Well, this has been a fascinating journey into a complex disease. We started with this hidden enemy, Porcine Proliferative Enteropathy, caused by that unique bacterium, Lawsonia intracellularis. We followed the decades-long scientific quest to even identify it, explored its really unusual way of manipulating host cells, making them grow instead of die. And we've seen how that knowledge led to better diagnostics and this crucial shift towards integrated control, with vaccination really stepping up as a key tool.
Yeah, and what's quite compelling, I think, is seeing how these global efforts to use antibiotics more responsibly in animals are fundamentally driving innovation in disease control. It's pushing the industry towards, more targeted, more holistic approaches, better biosecurity, smarter vaccination strategies. It really highlights this intricate dance between animal health, the economics of farming, and broader public health concerns in our world today. It maybe encourages you, the listener, to think about that constant learning curve in managing these complex biological systems in agriculture, and how understanding the details helps us appreciate the progress being made to keep both animals and our food system healthy.